Today we are very happy to have Dr. Alex Long from Hong Kong U to come here and to give us a talk related to MRI. So Dr. Long, he is now a research assistant professor at the Department of Electrical and Electronic Engineering at Hong Kong U. He received PhD degree from the same department, working under Professor Wu. So he is a top expert on imaging. So also, Dr. Long, during his PhD study, developed this optogenetic compatible MR technology for neuron stimulation to study the brain network. So he has published several articles in top journals like Nature Communication and PNAS. He has also received several for this excellent contribution to the community. He has also received several awards. For example, this Society for Magnetic Resonance Medicine Junior Fellow Award, and this I didn't know this long association.
Yeah, it's kind of like the overseas,
like Young Investigator Award. So his interest now is to use this developed technology and to study how the perturbation use this technology to look into the spatial temporal brain activity and the stimulation. And this kind of technology, neuromodulation technology, will have great potential for therapeutic application. And I think this is a good opportunity because we also have a lot of interest in developing theoretical framework for the neuromodulation. So I think after your talk, we will have some more discussion. Yeah. Yes. So you will talk about the window to understanding brain network and function through optogenetic functional MRI. Yeah, please,
okay,
Alex. Yeah.
Thank you, Professor Cho, for inviting me to give this wonderful seminar in front of the students of Baptist University, as well as like young faculty members of the university, as well as I just saw from CTU as well. Right. So I actually have spoke to Professor Cho for many years prior to this. I've always wanted to utilize some of our data to create a quantitative models to describe the brain networks and the properties, right? But today, what I would pretty much discuss or show to you would be our previous work, as well as also works from others currently using optogenetic function MRI to study brain networks and functions. So what is the goal of the contemporary neuroscience in general, not just looking at neuroimaging, is that we want to create a better understanding of how the brain generates different types of behavior. So you can think about different cognitive abilities to be able to read, think, speech, as well as the importance of sleep in regulating all these different kinds of functions, including the bodily functions, not just the brain, and also the different emotions that we have, right? So how do we link from the understanding of the brain networks all the way to our daily behavioral observations? And one of the two most utilized neuroimaging approach, especially from the lens of a magnetic resonance imaging scientist, would be to use diffusion MRI, as well as task-based or even resting state function MRI. So these two different modalities give you a window to look at the different components of the brain. So for example, diffusion MRI, we can take a look at the anatomical projections of whether it's the human brain or the rat brain. And famously, this is the backbone of the US NIH Human Connectome Project. And for the task-based and the resting state function MRI, so we are kind of more interested in the activations of the brain in response to, say, a stimulus. So for example, in this picture, first acquired in 1991, the first human fMRI image from the Harvard University, where they present a checkerboard visual stimulus, and then you're able to observe, activations in the visual cortex of the human brain. And probably about 3 years later, after 1991, and they managed to discover what we call resting state function MRI. So you do not present a task to the subject. We just ask the subject to rest or just lay down in the scanner with the eyes open. And you're able to detect this kind of homotopic, sensory, cortical networks, as well as a few other like attention networks, salient networks, default mode network. And also famously, one of the backbones of the US Human Connectome project, both the diffusion MRI and the resting state function MRI, just because it's very simple to acquire this in human subjects. But then there are limitations to the current approaches in able to, if we want to go further than just looking at the structure and some basic functions of the brain, right? We have not linked it to any behavioral outcomes or even getting close to understanding how those brain networks are related or associated to cognitive functions, right? Sorry. And this is predominantly because there is insufficient knowledge regarding the temporal dynamics of brain-wide interactions. So we're able to see those activations, but they don't mean anything if we don't really know what are their temporal dynamics, both spatially and temporally. And also, specifically, how do the long-range projections actually facilitate and govern all this kind of long-range or large-scale neuroactivity? So there must be some driving force or mechanisms that direct different kinds of activations and also involving different kind of networks for specific functions. So these are things that are still relatively unknown if you just used diffusion MRI and just task-based or resting state fMRI alone, right? So what would be the strategy to map or long-range brain network properties, as well as to investigate some of their functions? So as you know, like I'm a engineering, my background is in engineering, both my bachelor's as well as my PhD. So in our laboratory, we approach it as if like it's kind of a brain is like a computer, like a CPU circuit, right? So for example, we have this very simple architecture from the Intel Pentium 4 chip, right? And the best way to probe an electrical system, right, is to inject specific signals into different components of, say, for example, this chip architecture in order to study it, right? So the analogy here would be like, what if, right, we can have all this, with diffusion MRI, we are able to map all these very nice projections, as well as also from the Allen mouse brain project Atlas, right, where we have very nice mesoscale connections from even from the level of single neurons to different circuits, different cortical regions, subcortical regions, as well as even the cerebellum. And the analogy here would be, can we select different regions of the brain and we start injecting specific signals into different regions, and we look at observed, right, how the entire network behaves when each of these areas are perturbed, not simultaneously, but individually, right? And one way to do it, to look at the whole brain activity, as well as being able to modulate specific neurons in different brain regions, would be to use optogenetics, which will enable us to selectively target different neurons at different parts of the brain. So for example, in this case, I'm targeting the somatosensory thalamus of the rat brain. And then you're able to kind of implant a light fiber, and then you can design different kinds, or you can present different types of light pulses, whether it's a sinusoidal light pulse, a square pulse, yeah, as you wish, right? With up to like millisecond precision, that would be the fastest, you can say, channel kinetics for a basic protein of an optogenetic. And then we are going to use the functional MRI to observe the results of all these activations. So what happens when I activate the somatosensory thalamus, for example, right? Oh, and this is like kind of our facilities at Hong Kong U. So I will not claim that I would be the first to implement optogenetic functional MRI in rodents. It was actually a seminal work done by Lee and Karl Dezeroff in Stanford in almost more than a decade ago, where they also stimulated the motor cortex as well as the stimulation of the thalamus, and they're able to see thalamic activations upon cortical excitations, and then they're able to see cortical excitations upon thalamic excitation of using optogenetics and using fMRI to observe the, what happens when I present a light pulse at these different regions. So we took that approach and we applied it to different areas of the brain. So initially, we would be looking at, can we map long-range pathways, right? And maybe start to examine some of their functions with this optogenetic functional MRI. So I will show you some of our works based on the chronological order. So going from very simple to my PhD work, which is just looking at properties of a thalamocortical network and cortical network, as well as brainstem, thalamocortical network. And then finally, we took a step further to also look at the functions of, say, for example, memory functions through the hippocampal cortical and as well recently with the thalamocortical network. So in the beginning, you can see that this is a relatively old work. This was my PhD, the basis of my PhD thesis. So here we are looking at, can we map different types of long-range projections using stimulation with different or unique temporal properties, right? And as well, in the beginning, as I mentioned before, we stimulated the somatosensory thalamus, so you can see very nice expression at the VPM, as well as expression at the projection targets. So you can see the layer 4 and the layer 5 of the primary somatosensory cortex, as well as the secondary somatosensory cortex. Right? So this is very similar to what the Allen Atlas used as well in terms of the AAV virus to do projection tracing. But in this case, we are expressing a light-sensitive protein in addition to just being able to map the projections. And we did a simple paradigm where we stimulated the somatosensory thalamus at 1 Hertz, 5 Hertz, 10, 20, as well as the 40 Hertz, right? And what we observed in this particular study was, you can see that the gradual decrease of the stimulation frequency actually induces more spatial activations beyond the somatosensory regions, right? So as we know, the somatosensory thalamus is very well connected to the somatosensory cortex, so that is expected. But I think what was unexpected in our findings was when we went to the very low frequencies, kind of like almost slow oscillations, and we are able to observe bilateral, not just unilateral activations, but activation on the contralateral side of the somatosensory regions, as well as visual cortex and also auditory cortex. Right? So this, yes, Professor Joe.
Sorry. I asked, for different frequencies, why you use different frequencies? To avoid 1 Hertz is too long.
Yes.
Too much.
Yes, yeah. So because 1 Hertz is relatively a long pulse, so if 30% is about 300 milliseconds, so I think it's kind of unheard of to stimulate a neuron for such a long, especially excitatory neurons for such a long duration. Because typically, if you use a 300 milliseconds, then we found that you induce more than a single action potential, right? So if you want to keep it down to real 1 Hertz, which is you assume that you will activate a single action potential, then we need to keep the activation like shorter, the pulse duration shorter. And 100 milliseconds actually is not like the gold standard, just because like it's a balance between sensitivity in detecting the activations in MRI, as well as the neuroscience, you can say, right way to stimulate the neuron. So actually the best way is to do as short as possible, like 10 milliseconds, right? So we did it in 10 milliseconds, but the activations will be much weaker than what you observe here, just because activations in fMRI kind of scales with the duty cycle, yeah, on the linear, on the first order, right? Yeah, then we move on to not just looking at the Bow fMRI activations, but also to use electrophysiology to look at the underlying neural activity. It's mainly to confirm that these are all neural activities that are causing these activations, as well as we can also further, right, with electrophysiology, with better temporal resolution, we can look at the propagation of the neural activity across like a different pathway. So we can confirm that the ipsilateral stimulation of the VPM, it goes to the ipsilateral S1 first, and then it projects to the contralateral side. And at the same time, it has these very longer latencies when projecting from the S1 to the V1s. And then finally, you can see that the contralateral S1, V1 is also activated through the callosal projections. So these are things that we can kind of observe using multimodal approaches. And we also look at, we also demonstrate some flexibility in terms of maybe I want to look at, can the sensory processing be affected by your optogenetic stimulation, right? So kind of like, can we study some form of functions, not just mapping the long range pathways? Just because when we stimulate at 1 Hertz, we saw some strong activations in the visual regions, especially the visual cortex. So we look at, can the 1 Hertz stimulation affect just the basic visual, let's say, response to the visual stimuli? So in this case, say, for example, without optogenetic stimulation in fMRI, you can map very nice activations along the visual pathway. So we see the LGN, the visual thalamus, the superior colliculus, as well as the visual cortex. So this is a binocular visual stimulus. So both eyes are stimulated at the same time. And we can also present, so after acquiring this type of scans, we can now present and say, okay, when I turn on the optogenetic stimulation, right, what happens to this particular response? And we found that the superior, especially the superior colliculus, right, was affected the most in terms of the response, right, when we present the optogenetic stimulation. And we can also look at like what happens after we present the stimulus. Does the effect actually remain elevated, or does it go back to the original state as before? And we found that like not really, especially in the superior colliculus and in the especially in the superior colliculus, as well as the thalamus. There's some elevated activity, right, along the visual pathway, subsequent following the optogenetic stimulation. But there's just some very simple like concept, right, that we enable, saying that you can kind of study long-range pathways with these kind of techniques. And so, I mean, subsequently, I went to target a little bit more complicated region in the brain, which is known as the vestibular pathways. So if you don't know, the vestibular nucleus is actually connected to your ear. So there's like a ear, your semicircular canals with all the fluids inside to kind of allow you to, let's see. Yeah. So what are the essential vestibular functions, right? So number one, most important, balance and posture. So without the vestibular, and have you ever like taken a roller coaster and people say that, oh, after sitting a roller coaster, I feel nausea, right? I want to vomit. that's because your vestibular system is kind of firing like crazy because it doesn't know like it's your head. You feel like you're moving, but actually you're stationary. So there is kind of a conflict of sensory stimulus, right? And your vestibular system is responsible for, receiving all this information from your visual, auditory, your sensation, somatosensation, in order to tell you whether you are accelerating or you are stationary, right? Another one is ocular reflex, right? When you twist your head, your eyes actually automatically moves in the opposite direction. So that is another one. The sense of direction, velocity, and acceleration, of course, and of course, the reference frame. So actually your brain is calculating where you are in space at all times to tell you that you're standing and you're sitting so that you feel like safe, right? And if you were to try to study vestibular pathways in MRI settings, right? As you know, like these are the basic, a lot of the different fMRI experiments we have. Say, for example, in this case, the subject is presented with different taste stimulus, right? You can present different like taste, different odor, different type of smells, and then you can also stimulate the fingers, right? Finger tapping experiment, also very famous, even deliver some slight pain stimulus to the subject, as well as to the visual and the auditory. You can also present those kind of stimulus. Then you are able to map different types of activations corresponding to these different stimulus. But if you think about it, how would you do the vestibular stimulus? The only way to do it is you move the subject inside the scanner, right? You try to rotate the subject, whether in different directions at different angles. But as you know, fMRI is very sensitive to motion. You can't actually move the subject inside the scanner. So then you can't really fully study the vestibular pathways and the functions with the conventional fMRI. So what we did here is mainly what if we go look at the rodents, right? And we directly stimulate the vestibular nucleus, which is responsible for it's kind of like the nexus of vestibular sensory processing, right? And maybe we have a better chance of mapping the long range of brain networks that are responsible for vestibular processing. So we stimulate it. It's kind of like below the cerebellum at the hindbrain. And so we kind of categorized the results into like different pathways. So we know that like there should be activations along the sensory motor pathways. As you know, they receive inputs, right? And so as they may also send output to, say, the auditory cortex, the visual cortex, and the motor cortex, right? And as well as the somatosensory cortex. So these are all the expected regions that you would see, as well as the corresponding thalamus too. And we also found the activations at the ocular motor nucleus. As you remember, the ocular reflex. So this is also a region that is supposed to be projected, right, by the vestibular nucleus. So these are the known pathways. So the more subtle regions that like people don't really associate like vestibular nucleus to would be regions in the limbic areas, like the hippocampal formation, for example, subiculum, ectorhinal cortex, entorhinal cortex, dentate gyrus, right? So the vestibular system has always been more well studied for people who do space exploration. Right? So when they go to microgravity environments, they want to understand whether or not astronauts, right, when they are subjected to long-term exposure to microgravity, would their cognitive abilities be affected? Would their motor performance be affected, right? Because when you were born, you were more used to Earth's gravity. But then now if maybe in the future, you want to send humans to Mars, right? You are living in microgravity. Would that really affect your development of the brain, or even for an adult brain, will it affect, right, or change the physiology of the brain, right? And this shows that like the vestibular nucleus indeed would, right, affect some of the cognitive functions, especially maybe memory or even like cognitive performance. As well as other like more subcortical regions involved associated with hippocampal formation, like mammillary nucleus, the higher order thalamus, like anterior dorsal thalamus, right? So this is just like another example of how the optogenetic fMRI can be used to study more complex pathways in the brain, things that are not so well known, right, and not easy to be studied in the human subjects. And now we move on to a more complex circuit, which is the hippocampal to cortical network. So as we know, the function of the hippocampus is involved in the memory and spatial navigation, and the functions of the cerebral cortex, similarly, together the hippocampus also is very important for memory, attention, perception, cognition, awareness, thought, language, as well as the consciousness. So this is the kind of like the guiding principle, right, that we all know in the neuroscience. And what we are more interested here is that like how would the perturbation of the hippocampus affect large-scale brain networks? So as you remember in the beginning of my introduction, we look at resting state connectivity, right? So we're able to detect resting state in humans. Similarly, we can also visualize resting state networks in rodents, right? They're relatively similar, except maybe the rodent brains would not have so many networks as compared to the human brain, right? Say, for example, we have default mode in rodents, but we may not have like strong, say, salience network, right? Or more granular, different granular networks in the humans that we found compared to the rodents. So similarly, we took the same approach. The beginning for any of these experiments will always be to map the activations caused by the perturbation of the hippocampus. So in this case, we're looking at the lower frequencies of the stimulation. We stimulate the dentate gyrus, and we found that the visual regions are, again, very, very much affected by the stimulation of the dorsal dentate gyrus. So recently, we have another study, which I didn't show here, is that the stimulation of the ventral hippocampus. So the ventral hippocampus gives you a different activation. So we are seeing more of the frontal areas, frontal cortex with the ventral hippocampus, as well as the stronger activations in the entorhinal cortical areas, as well as the cerebellum when we stimulated the ventral hippocampus. But in this case, for dorsal dentate gyrus, we saw more towards the visual cortex and the superior colliculus. Right. But again, what we are more interested here is that if given the perturbation of the hippocampal cortical network at 1 Hertz, right, would that change the resting state, underlying resting state connectivity? So the easiest measurements for resting state will always be the bilateral networks that we always find. So if you look at the means before any perturbation, we are always able to map bilateral hippocampus network, bilateral visual, bilateral A1, and bilateral S1. And what you see that like during the stimulation, there's a strong enhancement of this connectivity, like bilateral connectivity, and it stays, right? It maintains its enhancement throughout probably up to 2 hours after stimulation as what we measured here, right? We didn't measure like further than 2 hours, but at least in this study, it shows that like there's a long-term enhancement of the resting state connectivity upon hippocampus perturbation. And similarly in this particular case here is more on the 40 Hertz stimulation. So instead of using a 1 Hertz stimulation, we use kind of a control stimulus, which only activated the like in the previous slide, only activated the hippocampus regions with the 40 Hertz. So we don't really see any enhancement in the V1, A1, or S1 resting state connectivity, but then we see some like general decreased, right, between the bilateral hippocampus for the 40 Hertz stimulation. And we're also wondering, right? So we see all this kind of enhanced resting state, long-term enhancement of resting state connectivity. So we did a simple behavioral experiments. So in this case, this is a novel object recognition. So the rodents, you know, they have a very propensity to explore new environments. So if you were to put them, say, for example, habituation in a test, we call this the home cage, right? So it's kind of like a square like environment. You don't put anything inside, they will just roam around maybe for 15 minutes. And then now you introduce like two objects, say 2 bottles, circular bottles, right? And what you will do is that like you will start to explore because there's something new, right, for it to go like play around with. So they will spend equal time between the two identical, we call it the square object, no, this circular object. And 60 minutes later, when we will test for the short-term memory. So if we swap one of the circular bottle with a square, right, will it be able to recognize that you introduce what we call a novel object, right, compared to a familiar object that it was exposed to earlier. And the rodents in the normal case will always go explore the novel object more because this is no longer exciting to the rat, right? You can remember. And if you were to do it 1 day later, and we test for the long-term memory, right? Now, the novel object in the short-term memory should become the familiar. And then we swap the familiar previously to now a hexagon, say, a hexagon bottle. And if it can remember, it would want to explore the novel object more than the familiar object, right? But for rodents, they don't have that kind of like capabilities like humans. They don't have the ability to remember for such a long period of time, especially after 1 day, that is able to remember that, oh, I already played with the square bottle yesterday, right? I shouldn't play with it anymore. I want to play with the hexagon right now. But for a rodent, it will not do that. That's why you can see that the discriminator index is hovering around 0. That means spending equal time between the object. But what we found is that when we present stimulation right here, right? When we present stimulation after the short-term memory test to the optogenetic animals, they're able to remember that yesterday I had a square bottle. Yeah. So this shows that like there are some long-term effects, right, caused by the perturbation of the hippocampus, especially when we look at the enhanced resting state connectivity. So this kind of like tells you that something is happening, right?
So when you make this?
Here. So after the short-term memory test, right, during this 24-hour period, we present about for 1 hour, right? 1 hour sporadically, we stimulated, say, like 1 Hertz for 1 minute every 5 minutes. Yeah, up to an hour. No, yeah. So the stimulation is when it's like we put it back to the home cage, and then we just like stimulate like the animal for 1 hour. Yeah, not continuously, but once every 5 minutes. Yeah, for 1 hour. Yeah. Then we take those animals and then we subject them to the test again, right? And typically for the control animals here meaning that they receive the same injection of the viral vectors, but just that they don't express the optogenetic proteins, right? And for those animals, you can see that like they spend equal time. Yeah. But then for the optogenetically stimulated animals with the effects, then they seem to spend more time with the novel object. They remember that I was exposed to the square the previous day, right? So this shows that there's some association between the enhancement of the resting state connectivity and some behavioral outcomes, right, that affects their memory performance. So this is the kind of like the one of our first study to kind of hint that perturbation of a specific area does have behavioral or functional consequences. Yes. The sample size here is about 8 animals for each. In this case, not very apparent. Yeah, it's quite consistent, right? We actually did like more, but we didn't publish those data. We stimulated at different points as well. We stimulated here, we stimulated here, we stimulated here, right? At different points just to see whether it's consolidation, but those are unpublished results. And in recently, which I think the paper will come out anytime soon, right? Would be we went back to the thalamus, right? But then now we are looking at the spindle oscillation activities and how it plays a role in associative memory consolidation, right? And in this case, just to show you how we initiate some spindle oscillations in the thalamocortical network. As you know, like spindle activities in the thalamus and cortex is kind of like what we call a waxing phase and a waning phase, right? So that's why they call it a spindle. So it looks like a spindle shape, right? And the way we try to induce spindle is to mimic its frequencies, right? As well as its duration. So we know spindle frequency is in between 7 to 15 hertz, and the duration is typically close to 0.5 to 3 seconds, right? And we can do that with probably 8 hertz at 24 pulses, which is gives you about 3 seconds in total stimulation duration. So if you were to look at the recordings at the cortex upon stimulation of the thalamus, is the yellow part would be the spindle activities. So these are the spontaneously recorded without any stimulation, right? So you can see this very clear that this is a spindle oscillations right here. This one lasts for about 1 second, and then it's about between 7 to 8 hertz, right? And these are the evoked activities. So we're using a slightly longer duration here, about 3 seconds, but with a similar frequencies, you can kind of see relatively same type of shape as a spindle. And of course, we believe that even if you use 1 second, you will also be the same, exactly the same type of response, right? The only reason why we chose longer duration is due to fMRI. Again, it's the sensitivity of detecting this kind of responses. If you're using a very short duration, then you would reduce the ability to detect some of these activations, right? But it's something that I think I would like to discuss with Professor Joe later, because in some of our newer studies, we're trying to say that this is not limited. The detection of the activations are not limited to the sensitivity of the bow. Can we actually use very, very short stimulus, right, in order to see some of these activations, which is more natural for looking at brain network properties. So we are able to detect activations in the sensory regions, but also a lot of the higher order areas in the brainstem and the basal ganglia, like substantia nigra, nucleus accumbens, right, hippocampus, and also the amygdala, insula, and the MPFC, the medial prefrontal cortex. So here, just to show you the kind of the most exciting part of the study, whereby what happens when we initiate this kind of spindle oscillations from the thalamus, right? We kind of designed a visual somatosensory associative fear memory consolidation. So if you're not familiar, it's relatively simple. You kind of associate the foot shock to a flashing light stimulus, right? In this case, it's a 10 seconds, 5 hertz light flash. So whenever you present the light stimulus, you present a foot shock as well. Then if you do this for I think 45 times, then the animal will start to learn, right, that whenever I present the flash, they would expect to have a foot shock. So they will freeze, right? They will not move. So then what we do there is this is the acquisition. So the extinction phase is whereby we test like the next day, right? if we just present the light flash without the foot shock, will the animal freeze? So if you look at the blue and the blue curve, for a normal animal in the beginning, it would freeze, right? But as you continue to present the stimulus without, then you will start to forget that, hey, it's okay, right? I don't really feel any pain anymore. So it would decrease generally, right? But for an optogenetically stimulated animal with evoked spindle oscillations, what happens is that it stays elevated throughout the entire extinction phase, right? So it shows that there is some effect when we present the spindle oscillations between the acquisition and the extinction phase, right? It shows that there's some enhanced consolidation of those memories that forces it to keep remembering that whenever I see a light flash, I should freeze because I should feel pain, right? Compared to a normal animal or even a sham normal animal, right? And if we were to put this animal into the functional MRI to look at what happens when we present this light flash, right? So for a sham animal, for a normal animal, we don't see significant differences, right? Except that you can see after the behavioral training, there's enhanced visual cortical activity. So as you know, there is a lot of studies showing that memory replay, right? The ability to remember memories based on your sensory experience is usually mediated by the cortical regions, right? It's kind of distributed from the hippocampus to the cortex. And you can see here that in the sham animals, the visual regions are enhanced, right? So this kind of makes sense. But then more interestingly is that if you look at the optogenetic animals, right, we find that in the post, it's not just the visual cortical regions, but we see very strong activations or enhancement in the periaqueductal gray, the superior colliculus, right? Even the somatosensory cortex, as well as the frontal regions, the frontal cortical areas, right? So showing you that the spindle oscillations thus recruit, right, stronger activations from regions that are responsible for associative field memory consolidation, especially periaqueductal gray, right? Has been known to be very well documented in the visual associative field memory. So that shows you right, the approach that, yes, I have a question. Oh, so that's a good question. I would stop short at saying causal, right? Because one thing you notice is that for any of these functional MRI experiments to be conducted, typically these animals are anesthetized, right? So we didn't go as far to say that they are causal, just because the conditions when we perform the behavioral experiments and the conditions that we perform the fMRI experiments, the only difference is the anesthetic agent, right? We use isoflurane to anesthetize the animals for our fMRI, right? Which is why I think I would kind of say some of the approaches that other people are using, right? Which is to perform these experiments in awake animals, right? So I think the field is moving towards awake functional MRI for rodents in order to get a better, you can say, to better associate your experiments between the behavioral task and maybe even do the behavioral task inside the scanner, right? While the animal is awake. Yeah. Then I would go and say that yes, it's going to be very close to a causal, right? Yes.
You have this stimulation of
Yes.
you have control.
Yeah, just one group compared to control. So the thing that we didn't do was to, what if we evoke activities that are not spindle, right? Say for example, we use a 20 hertz, say for example, stimulation at 3 seconds, right? would it also would it like give us a negative results, or that it also gives us these like spindle oscillations? Because the only like it's unpublished, but the only like evidence that we have is that when we do the spindle oscillations, now we are doing with the resting state connectivity, only the 8 Hertz affects the enhance the resting state connectivity. But if you use 20 Hertz with the same duration, it doesn't. There's no effect to the resting state connectivity. So my hunch is that like if we were to do the behavioral experiment, it likely won't like show this kind of enhancement to the memory consolidation functions. Is to the excitatory neurons of the thalamus. So it's chem kinase 2 alpha. So it's all excitatory neurons in the thalamus. Yes. Yeah, because we're kind of invoking, we try to stimulate spindle oscillations along this like a thalamus and cortex network, right? So we can do the excitation of the excitatory neurons. So similarly, you will see that like there are also very similar approach taken by other groups in the world, right? And that includes, this is from UCLA, they're looking at cerebellum, right? Cerebellum networks, trying to stimulate here, and then looking at, do I see any activations in the frontal regions of the brain, as well as, this is also from the Stanford group that did the seminal work. Here is looking at the, if I'm not mistaken, it's a central thalamus stimulation, right? And then showing different kinds of like dynamic response as well, right? So these are all the like the earlier studies. You can see that is always using this technology to map long range or different types of different networks. Similarly here, they extend it to neurotransmitter systems as well, right? Not just excitatory neurons like we did in the previous studies. So here they're looking for a serotonin system, right? Stimulating the dorsal raphe nucleus, the 5-HT neurons. And I think for this from UNC, they are like North Carolina, they're looking at, if I'm not mistaken, this is dopaminergic neurons, yes, in the substantia nigra. Yeah, also looking at the different response, right? And then more recently in 2023, you can see like people are moving towards more complex interpretation of the optogenetic functional MRI data. So this is like trying to use some modeling, Bayesian, I think it was a Bayesian static dynamic system model, right? To look at the brain networks upon optogenetic perturbation of the insular cortex. So they're looking for like different states, different latent brain states that transition, right? Before stimulation and upon stimulation and after stimulation. So they can find that like during the on state versus the off state, they are kind of switching between different areas of the prefrontal regions. Sometimes some are more weighted towards the prefrontal, some are more equally distributed, right? Maybe in between, say, in between stimulations and in between after stimulations. So showing that we can use some quantitative models to better describe the temporal dynamics of how the brain networks are changing upon perturbation, right? Not just a single measurement like we did in the past, which is like 6 minutes, every 6 minutes we have one measurement. But in this case, the resolution is much higher. They're looking at like a sub-second to a second resolution of how the connectivity is changing, right? In response to the stimulation, as well as after the stimulation. We're also very interested, not just during the stimulation, but we're also very interested in what happens after that, right? We want to observe whether the effects are long-lasting or the effects are just very acute, right? This gives us a better understanding of what is actually going on, right, to the networks, right? And then These are like our collaborators in the Shanghai Institute of Neuroscience, right? So Dr. Liang, they are the pioneers of rodent awake function MRI there, both using optogenetics. In this case, like this is the optogenetic stimulation of the MPFC, right? Both in the awake state versus the anesthetized state. And then they also do a lot of like awake using, you can do auditory stimulus, somatosensory stimulus, you know, all kinds of like task fMRI in the awake animals, right? Yes. Yes. Yeah. But I think like for rats will be much tougher. Yeah, because you know rats are generally stronger, so they would try to resist like putting them inside the scanner. So I think like they're kind of like migrating towards mouse, which is more manageable. Yeah, mouse are smaller, easier to immobilize, right? Inside the scanner compared to like rats. Yeah. I think now with like better MRI hardware, because we don't have a cryo coil in Hong Kong U. I think in Guangzhou they have one. If you're using cryo probes, then you can improve your SNR by at least 5 times compared to non-cryo probe for mouse, right? Then they can do like very nice, like for example, these are mouse fMRI. This is done in Stanford, right? With Gary Glover and also with Karl Dezerov. So they're looking at reward stimulation here, stimulating the striatum, yeah, to look at how, and doing some behavior experiments inside the scanner while awake, right? Your data, what is the temporal resolution right now? The acquisition is about 1 second every data point, right? We can go faster, like with like newer image acquisition and reconstruction, we can go down to 500 milliseconds, yeah, without significant loss in SNR, right? But there will always be some penalties, like in terms of like sensitivity in detecting the bow activations, yeah. And so what would be the future, like before I end the talk, what would be the future of this like probing long range, like brain networks, right? So the goal is not just to observe or visualize all these kind of nice images, right? The goal is that like, can we start to create quantitative models to describe, right, all these different like activations that we observed and trying to link them to, you know, the final output, which is like motor control, motor execution, as well as your behavioral modulation, right? Can we bridge the gap between these two different worlds? And I think the most important why we want to bridge this, right, would be neurotherapeutics, right? In order to design better therapies for challenging neurological disorders, you would need to have better treatment strategies, right? Currently, we are always relying on the pharmacological pathways to solve, let's say, Alzheimer's, to solve Parkinson's, right? And I think the only current clinical method to solve like Parkinson would be, I mean, the most effective one would be deep brain stimulation, right? That's the only like we can say neurostimulation therapeutics that is like FDA approved and commonly, you know, done for patients suffering from tremors is DBS, right? Other than DBS, there's nothing else, right? There's no other ways to treat like, say, for example, depression, anxiety, right? It's all mostly through like pharmaceutical methods. But that's because we don't really understand the dynamics of the brain networks underlying it. So these are always network issues, right? In the larger scale, it's not a singular neuron or a singular region of the brain. The brain is connected after all. So it's a network issue. But the reason why we are unable to solve them is because we currently do not have a very good quantitative description of brain networks, right? And how it relates to the functions that we want to solve, right? But if we are able to do that, then it would improve our neurotherapeutic strategies for the future. And we are able to go to like different brain areas, maybe do some brain stimulation. I think a very good example at the moment would be MIT, right? They have these like goggles that you wear for Alzheimer's patients, right? They have this study in the mouse done where if you stimulate the mouse for every day for like maybe 40 minutes with like 40 hertz flashing light, because they say that like it can induce gamma oscillations. Then they found that like the A beta plaques, right, was reduced. And they found that there's elevation of symptoms as well after exposure to light flashes. And they're currently doing, like, as far as I know, they're doing some clinical trials from a company that they created, right, out of this like study for subjecting Alzheimer patients to light therapy for every hour up to 2 weeks, I think, and to see whether or not they have improvement in their symptoms, Alzheimer like symptoms. So this is also a very good like example of like what you can say the neurotherapeutic strategies would be like, right? Other than just using pharmaceutical methods, is to do some neuromodulation to improve your symptoms, right? And before I end, I would like to thank my mentor, Professor Wu, right, who have worked very closely for many years, as well as you can see the many currently, like some of them are already PIs and working in industry, who I worked with in Hong Kong U in the lab, as well as the many collaborators that I have the pleasure of meeting and also discussing my work with, right? And last but not least, the funding support from RGC and the Lam Wu Foundation, as well as the Guangdong Provincial Key Brain Research Project, right? That made all these studies possible. Yeah. So I thank you for your attention.
Excellent talk. Thank you very much. Now, questions, Adam?
So, yes, thank you. This is very sort of tantalizing. There's like, I'm sure a lot of us would love to get to work with some of these data, but wondering, so at 1 second time resolution, that's a lot slower than the rate at which neural impulses are actually transmitted through the brain. So at raises, so that leads one to wonder, okay, so what do the changes represent? Like a sort of new equilibrium state, a new sort of steady state that's introduced? At what point does, like, how much indirection is possible from one time slice to the next? So, and how would you sort of represent, basically, how would you approach that? What would you say is it actually means if you see stimulation at if stimulation happens at one time point and you see areas differentially activated at another?
Yes, that's a good like question. So, I mean the issues with like functional MRI data, like as you pointed out, has always been much lower temporal resolution than what you would be able to achieve with, let's say, electrophysiology, right? So I think the key here is that, like, I mean, I didn't really like focus too much was the multimodal measurements. So for actually every of our MRI measurements that we do, we actually did a complementary electrophysiological measurements at the critical regions that we observed are changing or are responding to the stimulus, right? So with the electrophysiological recordings, then we're able to really look at the direct neural activations that either changes or being modulated or in response to that stimulus, right? Then we're able to extract or can go further in terms of examining neural mechanisms, right? So the fMRI part, the good thing is that it's kind of like a large camera, right? So we're able to observe the whole brain activations, and compared to electrophysiology, then with electrophysiology, it's kind of like we use the fMRI to guide it. And we put the electrodes at those areas, and then we do further examination deeper, because there's only so much that you can kind of extract from the fMRI data. We can have very nice changes of connectivity, but they don't mean anything, right? If you don't observe like direct neural activity, or you can't pinpoint the exact neural mechanisms that
Okay,
changes, the network.
yeah, so that's an important point. That
Mm,
really lends a lot of credibility to those results. But then it sounds like you're working with implanted electrodes then?
yes.
Yeah, now that could be harder to do with humans, of course. So
Yeah.
do you think it would be possible to do something similar with EEG, for example?
Yes, it's possible. I think the only issues would be like I think for humans, it's just that there is like with EEG, it's kind of like heavily weighted towards the cortical regions, right? So the subcortical regions are usually kind of like neglected in some ways, right? We kind of still see some you can say average signal from the subcortical, but we cannot pinpoint exactly which subcortical region, right? In the human brain. I mean, the key there would be, can we innovate technologies? Maybe one day, right, we can record neural activity deep inside the human brain just from the surface, right? That would be the challenge, right? For like I think the Brain Initiative in the US, they are trying to promote this kind of advancement of neurotechnology, right? It's possible with EEG, I would say, to do some of these studies. I've seen they're using EEG in humans, and then maybe you have a chance to do invasive recordings in patient groups, right? There are some groups in the world that they implant electrodes in like epileptic patients. So because it's experimental research, they're able to record from 1 or 2 subjects from deep brain, right? In order to understand better and try to relate it to the animal studies that you can do invasive recordings. So that's one way.
Yeah, so actually, an interesting thought. Now, though, since you bring it up, there are actually areas of the brain where fMRI is a lot less reliable, where fMRI is a lot noisier in some deep brain regions. So I'm wondering how you can address that in these studies. Like, you have to just ignore or take with a grain of salt what you're seeing in some of these areas, or is the quality, well, in mice, I suppose it's smaller, so less noise, it's easier to penetrate those reads, but thinking that's another area where there's a bit of a hurdle to scaling this process up to humans. And I'm wondering whether you have any thoughts on how to handle that.
I think the key challenge for humans will always be invasive experiments, right? Especially like normal human like subject. I think no one would. I think there's something that we would never be. I don't like to say never, but I think very difficult to have ethical approval to really do some like invasive procedure in normal human subjects, right? But like the only way to resolve this is to have better imaging technologies, that you can go for like direct imaging of neural activations, right? Deep brain. I mean, MRI, of course, it's not just functional MRI, right? But they are, the holy grail for functional MRI actually is what we call non-bowl functional MRI. So what we observe is all secondary, kind of like secondary activations, because those are like changes in the blood oxygenation levels. So we call it blood oxygen level dependent signals, or BOL for short. But the holy grail for fMRI is actually non-BOL imaging. That means like, can we use MRI to image neural activity, right, directly? Right.
If it comes to
Yes,
invasive versus non-invasive, there's also a sort of middle ground in the world of PET and radio imaging where you're injecting something but not actually opening up the skull. So I'm wondering whether you could combine some of this work with PET or other radio imaging.
yeah. So So like I do know of a colleague, right, at I think at MIT, they're doing this, what we call molecular functional MRI. So they're using like agents, like different probes, right, that are tagged to say either calcium, right, they're tagged to maybe the sodium potassium channels, they're tagged to aquaporine channels, right? Then what you're measuring is no longer the blood signals or the neuroactivation. You're actually looking at the molecules, the probes, right? So that could be one to bridge the gap between like say non-invasive and invasive, right? I think the only issue there is like, can they get those agents to be approved for human use, right? So right now they show it's very good for very good results in rodents, no problem. Primates as well, no issues. But I think the next step would be to get that approved for, injection or like injection into like humans, right? Because the only contrast agent right now is for MRI is like gadolinium, but those are like for structural tumor imaging. They're not for like functional imaging per se. Yeah.
Well, I mean, it's usually used in a medical context because that's where there's
Yes.
a need that justifies the risk of radiation
Yes.
exposure. So,
Yeah, right.
yeah,
Yeah, so as long as the probes are safe enough, I think once you clear that hurdle, then yeah, you can do like very direct molecular imaging, right? Molecular functional imaging in the human brain. And that would solve like, that will give you an extra, you can say specificity, right? That you do not get with conventional functional MRI or like, to resolve that invasive issues.
yeah, I think I've kind of monopolized it for a while. Let's
Sure.
Hey, I follow up on
Yes.
the data you showed in one of the slides is the alpha simulation. You talk about the pathway, and you mentioned some millisecond of how you obtain that.
So you mean
I
the,
see on the slides, you see kind of a activation delay with certain 30 milliseconds.
oh no,
It's
those,
just a piece of-
those are electrophysiology for the latencies. Yeah. Well, I think it's tough to look at like a millisecond for fMRI unless you acquire at millisecond time resolution as well. But then there's a dilemma there. It's because like in the community, as we know, the blood flow, right, is not that fast anyway. It's about, I think the fastest ever recorded is about 100 to 200 milliseconds in terms of the vessel dilation and constriction, right? So they were saying like, would it be meaningful to look at anything below, let's say like 100 milliseconds or 200 milliseconds for like bowl imaging, right? That's a debate that I think they've been debating for many, many years. Yeah.
From us, we go to the recognition network. You see once you have activation, with all this, so even they think this measurement accuracy of a second. There's already go through the group signal.
Yes.
They're hard to associate with the underlying.
Yeah.
Yeah.
Yeah. So I would say like the best use I think the future would be really like using multimodal data. So that means you have fMRI, which takes care of the macro, kind of like macro scale. And then maybe you have electrophysiology or an optical imaging technique that would take care of the meso scale, right? And you try to acquire them simultaneously at best, right? I know it's technically challenging, or even try to acquire them at identical conditions, right? Then you can start to like bridge or kind of create a model across different spatial and temporal scales, right? Then that would take care of like you can say a overview of the entire network, right? At micro, meso to macro. Yeah.
One of your work show that the low frequency activity may drive the brain wide resting state connectivity.
Yes.
I also saw the similar results in other places. So I'm always wondering, why is it the low frequency activity? Are there any computational advantages of low frequency activity? Because I think whether the low frequency activity is a little bit too slow to integrate multisensory information.
So I think the good thing with these slow oscillations, I mean, if you just look at it from electrical signal point of view, right? I mean, the AM versus, let's say, the FM, like radio, correct? So as you know, like AM radio uses much lower frequencies than FM, like a radio. So the good thing about slow oscillations as a wave is that it can propagate much further without loss of information compared to, say, higher frequencies oscillations, right? So you kind of have like more data loss with higher frequencies than with slow oscillations, right? And also with slow oscillations, actually the brain requires like a temporal window of integration, right? It's like there's like a window time whereby like, okay, this is where all the important computations can happen for this particular like a function. If you were to use the high frequencies to do that, then you also have like, I would assume, would have lots of signal lost or like failure rate in terms of your being able to do some integration between different sensory inputs, right? So with slow oscillation with a longer envelope, right, then you have more degree of freedom in terms of integrating different inputs and also to execute different outputs to longer ranged regions, right? But this is purely me speaking from like from an electrical engineer, right? When I deal with like electrical signals, but I would assume that that would also be quite applicable to the electrical signals in the brain as well, right? Ultimately, it's just different medium, maybe slightly different like computational like circuits, but it's still an electrical signal, right? So the properties would, I would assume, would be relatively similar. Yeah.
Could I go this
Yes.
I did slow oscillation and fast in terms of local circuit activation. Fast signal may also introduce the activation.
Yes. Yes.
But this slow oscillation may couple with certain cortex wide and larger to a certain level of some slow prosthetic process, which may have certain difficult slow of or some neural modulation. That's they mentioned that the articulation the functional pattern will make different for our necessarily
Yes.
related to certain that's like that. Your data going like lower they become
Yes. Yes. Yeah. So I mean, there's always that like debate whereby, say, for example, for memory functions, right? Like at certain time points, I mean, it's more like weighted towards the hippocampus. And then they say that like for longer term memory consolidation, then the hippocampus will start to distribute the memories to the different cortical regions, right? And I would assume that like that particular distribution and integration would involve different types of oscillations. Yeah.
actually
Yes. Yes. Correct. And then maybe like, I mean, they're hinting, people have hinted that like the slow oscillations are very good for like, or at least seems to be very important for this kind of distributed activities to longer range pathways further away from the local regions, from the local circuits, right? At least those are like from just direct observations. And yeah, also from sleep and also optogenetic perturbation at different brain states. So there are some studies that like to stimulate at the up states of the slow oscillations versus like in phase or out of phase, right? And they found that like if you were to perturb the network in phase with the slow oscillations, the effects are much more pronounced than if you were to stimulate out of phase with the slow oscillations. So that kind of gives us an idea that slow oscillations possibly would involve this kind of like more network-like properties compared to the higher oscillations, like higher frequency, which is more local.
Also want to drop this discussion because in physics there is phenomenon called resonance. Basically, it tells you that if your input frequency is in line with your intrinsic frequencies, the response will be somehow maximal. So maybe I think I feel like maybe in the anesthesia state the brain may operate at somehow low frequency regions, and in that time the low frequencies will somehow match with your intrinsic frequency of brain state. Then the low frequency stimulus can probably level. When maybe in the awake animals, in that place maybe we need somehow high frequencies to induce such similar phenomena.
Yes, I mean that is like one of those like most like very well documented, say, electrophysiology in terms of the power spectrum, right? When you're sleeping and when you're resting, the lower frequencies dominates, right? And then as you start to transition to an awake state, then you start to observe that your neural activity is going higher and higher frequencies, right? If you were to look at your recording. Similarly, if you like under anesthesia versus no anesthesia, then you see much more pronounced high frequency activities in non-anesthetized animals than say anesthetized animals.
Well, maybe we can take a break and then